Hi, I'm Caroline DeJager.
I'm Kayla Purcell.
And I'm Hannah Wilding. And we did research on the differential expression of appetite regulating genes in avian models of anorexia and obesity. So for the background of our appetite study, it has been long known that the hypothalamus is used in controlling commands of hunger and satiety in animals. Within the hypothalamus, there are separate nuclei, which are aggregations of neurons that control this hunger and satiety. The first is the arcuate nucleus, which is a nucleus at the base of the hypothalamus. Next, we have the ventromedial hypothalamic nucleus, which deals with satiety and this prevents the animal from starting to eat. This can be considered like a parking brake. Next, we have the dorsomedial nucleus, which also deals with satiety. However, this deals with meal-ending satiety. This can be considered a brake. Next, we have the paraventricular nucleus, which deals with stress. But it also has neurotransmitters that deal with hunger and satiety, so it deals with both of these aspects. And finally, we have the lateral hypothalamic nucleus, which is a collection of neurons that doesn't really fit elsewhere. So since it is associated with hunger, it is grouped into the hypothalamus. So here you can see we have the arcuate nucleus, the ventromedial hypothalamic nucleus, the dorsomedial nucleus, and the paraventricular nucleus. And those are sort of all oriented like superior to one another. And then the lateral hypothalamic nucleus is kind of grouped off on its own to the side. Next, there are chemicals that work, chemicals and neurotransmitters that work within all of these nuclei, and so they also control hunger and satiety. So for mesotocin, it inhibits food intake in chickens and mammals when it is centrally injected. And for orexin, it has neurons in the LHA that play important roles in motivated behaviors such as feeding and drinking. Signaling of this orexin increases the sympathetic drive, and it is a neuropeptide that regulates arousal, wakefulness, and appetite. Next, we have ORXR2, which is a G protein coupled receptor for orexin. NPY is a 36 amino acid neuropeptide that acts as a neurotransmitter and influences food intake, and it can be shown off to the side on the right. NPY-R5 is an NPY receptor. MC3R is a melanocortin receptor that is activated by members of the melanocortin family. And AGRP is co-expressed with NPY, and it acts to increase appetite. It is one of the most potent and longest-lasting appetite stimulators.
So these definitions were most prevalent in the research. An inducible gene is only activated in the presence of a certain molecule. CFOS is an example of this. Orexigenic refers to appetite stimulants. Anorexigenic refers to appetite suppressants. Refractory means that something is resistant to a process. This is relevant because the LWS was refractory to food intake induced by the injected NPY.
Okay, so this study was performed by Dr. Klein and Paul Siegel, who's shown right here, and it focused on this study using chicks as a model. So the chicks were classified as either low or high body weight, and they were grouped split up two ways after that into LWS fed, LWS fasted, HWS fed, and HWS fasted. And these chicks were inbred for 57 generations. So one of the methods that was utilized in this research study is Tukey's statistical test. And the graphs were broken up into 4 different parts. The LWS, HWS, and each one had a fed and fasted bar. And the letters above were used to determine the statistical significance, and the different letters show that the results are statistically different from one another.
Okay, so now we have the results of our study. And first of all, we have found that there is a greater sympathetic tone in the LWS, which is the low-fed chickens, than in the low-weight chickens, and the HWS, which are the high-weight chickens. This is due to the fact that there's more mesatonin in the PVN, more ORXR2 in the VMH, and more orexin in the LHA. And that is all LWS comparative to HWS. So in terms of the mesatonin, it inhibits food intake when it is injected and is twofold greater in the LWS than in the HWS. And for orexin, the mRNA in the LHA is almost 8-fold more expressed in the LWS than in the HWS. And this is the largest difference observed in our entire study. And so the injection of orexin leads to increased sympathetic drive. And so in terms of the increased sympathetic tone, this leads to more flight or flight response because it deals with the autonomic nervous system. And this also leads to slow digestion, which decreases hunger. So that explains why these low-weight chicks have anorexic tendencies. Next, we have the MPY signaling is active in the LWS, but is overpowered by the anorexigenic tone. This is due to more MPY in the ARC and DMN and more MPYR5 in the LHA, PVN, and ARC. The MPY is threefold more greatly expressed in the LWS than in the HWS in the DMN, and upregulation in the LWS, or it is upregulated in the LWS, but is not in the HWS in response to fasting in the ARC. So this is due to an attempt to compensate for the anorexigenic tone. There's also more MPY expressed in the LWS, but the actual amount of food obtained by the low-weight chickens is way less than the amount of food obtained by the high-weight chickens, and so this supports the fact that even though there are orexigenic signals in these low-weight chickens, the anorexigenic signals overpower these, and the satiety-inducing mechanism is very reactive. And next we have the melanocortin signaling is much stronger in the low-weight chickens than the high-weight chickens. This also may support the anorexigenic phenotype, and the greater expression of this is due to greater expression of MC3R in the nuclei and greater expression of AGRP in the arcuate nucleus. So the MC3R is more greatly expressed in the arcuate nucleus and is also upregulated in the DMN by fasting for the low-weight chickens. The greater expression of MC3R shows that there is stronger melanocortin signaling in the low-weight chickens, and these melanocortin receptors are receptors for alpha MSH, which is a very potent satiety factor. So this can explain the lack of appetite and the different body composition of the low-weight chickens, because if a satiety factor is present, these chicks won't be eating as much. And lastly, the study wasn't largely determined to have huge differences between fasting brought about between the fasting and fed. It was more done between low-weight and high-weight chickens, but there were some differences found by fasting. First of all, expression of MPYR5 in the PVN is dampened by fasting, and in a previous report, this is supported too, because it says that restriction of calories increases MPY release, which decreases the expression of MPYR5, and so this can balance out to normal caloric intake. There's an increase in the MC3R in the DMN and an increase in the MPY in the arcuate nucleus in the low-weight chickens, but not in the high-weight chickens, and this can explain compulsive feeding in the obese chickens.
So one of the most important parts of animal research is doing it in an ethical manner, and we know this was done ethically because the protocol was approved by the Institutional Animal Care and Use Committee at Virginia Tech. B-actin and leptin were both studied in the research, and this is important because they're also found in humans, and all animal research should go to a greater cause to understand more about other species. And mutations in B-actin and leptin have shown devastating results in people, and therefore it's important to study these both. So some things to change for future experiments is oftentimes inbred organisms end up with deformities and other issues. For further research, the testing of unrelated chicks would reduce the number of problems that go along with inbreeding, such as reduced biological fitness. When studying the LHA, the orexin was 8-fold higher in the LWS than the HWS. However, when the hypothalamus was studied, the orexin levels were higher in the HWS than the LWS. This makes us wonder about the CART because it's a chemical that leads to increased food intake. Levels of CART were higher by 4-fold in the PVN of the HWS than the LWS, which explains the compulsive eating by obese chicks. However, we're interested in whether CART and other chemicals follows the same trend as orexin. So in this experiment, a cryostat was used. So another thing that we could change is maybe using a vibrotome or another instrument to cut the slices. Vibrotome attempts to keep the samples alive, which reduces the number of broken cell membranes, which is important in these experiments. And then there was a stressor in the early post-hatch period of chicks, which could have changed the results. So this research was very intriguing to us, which led us to look at other questions that we could do more research on. So one thing that was mentioned earlier was the levels of neurotransmitters in individual nuclei compared to the entire hypothalamus. And another important question to ask is how can this information or how can this study be extrapolated to other species, particularly people? So we wanted to look into how the findings and results of this research can help find new treatments for obesity and anorexia in people.
Right, and here are our citations for our pictures and for the rest of our research study. And thank you so much for listening.